Here's a picture of a human brain from the top and from the side. It weighs about 3 pounds and contains approximately 100 billion neurons with an average of 7,000 synapses per neuron. You can see it's divided into two mostly symmetrical halves, which are called hemispheres. There are also grooves or folds called sulci, and each bump between the grooves is called a gyrus. Everything you can see from the top of this intact brain is some kind of cortex. On the bottom of the side view, you can see a part with an even higher density of folding. This is the cerebellum, which plays an important role in coordinating our movements. If you turn the brain over, you can see a cylindrical bundle entering into the bottom of the brain. This is the brainstem, which in your body would connect to the spinal cord. There are many more structures within the brain that you can't appreciate from this external view. Here we have a picture of a slice through the brain, allowing you to see some of those internal structures. The first thing you'll notice is that there are two different colors of brain tissue. The darker parts are called gray matter, and that's where the cell bodies of the neurons are located. The lighter parts are called white matter. They're mostly composed of axons running from one brain area to another. And the fatty myelin coating around the axons is what makes this portion of the brain appear lighter. You can see there's one continuous folded layer of gray matter all around the surface of each hemisphere. This is the cortex. There are also organized neuronal structures in the interior of the brain. As a group, these structures are all referred to as subcortical, below the cortex. And this includes areas like the hippocampus, the thalamus, hypothalamus, amygdala, and basal ganglia. Although the cortex looks like one continuous wrinkled sheet of cells, in fact, it has many different areas within it. These areas differ from one another in what other brain areas they connect to and in the properties of the neurons they contain. Here, some of the brain areas in the human cortex are labeled with numbers according to their Brodmann area. This is a scientist who labeled the different brain areas, but many of these areas also have other names, often descriptive of either their function or their physical location. One of the first indications that particular brain areas might be associated with specific functions came from electrical stimulation experiments. As early as 1870, two scientists named Fritsch and Hitzig demonstrated that electrical currents applied to the surface of the cortex could evoke movements. These movements were generally on the opposite side of the body from the cortical site being stimulated, and small movements of the electrical contacts activated different but reproducible muscle groups. In the mid-1900s, experiments in conscious human neurosurgical patients allowed evaluation of perceptual as well as motor effects of electrical stimulation in different parts of the brain. For example, patients reported seeing spots of light during stimulation of the occipital cortex near the back of the brain. We can also see specific deficits in patients that have damage to one particular part of the brain. Damage could result, for example, from a stroke or a traumatic accident. As one example, damage to the region known as Broca's area impairs speech production, but not understanding. We'll talk more about some specific brain areas and their functions throughout the rest of this module.